And unmute my face here. All right, so finally, we're getting to the main topic of the seminar here, resource competition theory. We spent a little bit more time on Lotka Volterra than I anticipated, but finally we're here. But just as a refresher, why are we here? So this is how we wrapped up a couple classes ago, talking about pros and cons of Lotka Volterra competition. And not to trash it because it's still like super useful. But the good thing about it is like really easy to understand, illustrates all the different outcomes of competition. It's pretty general, right? It's not really making a lot of assumptions about the particular mechanisms of competition. But the bad side is mostly about like the interpretation of the parameters. So there's really you can't go and measure those parameters in Lotka Volterra competition without doing competition experiments. It's not clear how those parameters, competition coefficients, depend on environmental gradients, and there's a lot of them, and how are they related to each other? Not so clear. So these are the things that we're going to try and solve with the resource competition theory. Wait, Leon, you want to talk about your thing now?
Now or after your lecture?
Maybe, all right, maybe at the end, but
Okay.
let's not forget. Okay, so, all right, so question is, what is a resource? And maybe just to get people like slightly interactive today, because there's gonna be the last time, hopefully, that it's gonna be like one full session of lecture, and then we'll move into like paper discussion mixed with a little bit of lecture. But I'm gonna break into the breakout rooms right now, and you can discuss two things. What is a definition of a resource? And then what are some examples of resources? And we'll just do that for 3-4 minutes here, and then we'll come back. So make a few of these things. Okay, perfect. So see you in a few minutes. All right, so be all nice discussions. We had a nice one here. Who wants to suggest a definition of a resource? Anyone?
Yeah, our group kind of came to this idea of a resource. It's something which is like causally relevant to survival and fecundity, and something that is consumed, and something that is relevant or that improves survival and fecundity only insofar as it is consumed.
Okay. Fecundity. Oh, you said fecundity, not reproduction. I'm a mile asleep.
Consumed or used?
All right. Should never do that. Madeline, what do you think?
I think going to what Matt was saying about consumed or used, our group was talking about not just something that would be obviously consumed, like food, but we're also talking about something like space, which maybe is not consumed but is used.
Okay.
Yeah.
All right, so I'm gonna move to the next slide because I already wrote my own definition here, but which is pretty similar. Increases growth. I would say, all right, yeah, growth, population growth, and it's consumed. So examples, I heard a couple already. Food, space. What other examples y'all got? And you don't have to raise your hand, you can just jump in.
Light.
Light? Okay. Yeah, Eleanor?
Right. So, going back to the is consumed thing, so with the is consumed thing, one of the things that my lab studies is shelter. We call them resources, for insects. Like maybe they need, like bumblebees, for example, will nest in old mouse rodent burrows, or need certain types of soil that they can dig in, or holes in trees of certain sizes. So I'd advocate for not just consumed.
Okay, so consume / use, that's good.
I would say maybe nutrients, sort of like food, but it's more general.
Yeah, nutrients, classic. Probably what we're going to talk about the whole time today. Any other out there? Wait, so what was that, like tree holes? I heard that in Eleanor's.
Thomas brought up a good point in ours about whether like for sexual
Mates.
selection, yeah, mates. So is that a resource?
Okay. All right, we'll have to think about that one later, but maybe, maybe.
Think of it as a resource.
Here is something I thought about a couple of times. Is air or oxygen a resource? You clearly need it and you consume it, but there is basically never any competition for it. Maybe there are in some weird systems, but
in the ocean.
Yeah, what was Jen?
Sometimes in the ocean there are competition for oxygen.
Okay,
Yeah,
so
I guess in
there's
some systems. Yeah.
okay, so maybe we need to add something. So what you're saying is that it's consumed and used and actually depleted, maybe?
but there's some limitation to its availability, I guess, is the crux.
Limited availability. Yeah.
But does a resource by definition have to be limited? You know, if it's something that an animal
Non-limiting resource. Yeah.
Yeah.
Okay. All right, this is a question. I think we'll leave this one for the philosophers. All right, other thoughts? So right, so oxygen's an interesting one because usually like we consume it, but like usually there's more than enough, but not always, right? There's like oxygen minimum zones and so on. What about temperature? What do y'all think about temperature? Is it a resource?
I think if it's tied up with like space, if we think about thermal refugia, in that being linked to, you know, shaded areas or something like that, maybe, but it's kind of a byproduct of space.
Mhm.
Yeah, that came up in our group too, and I was in the camp that it wasn't, but I agree that in this context, it could be like but that's kind of like I think of that as like a shelter. But
Right.
like the temperature dependence of metabolism, I think that's, I don't know.
Well, why
Yeah, I
not?
generally don't think of
What's the
temperature.
problem with, I mean, increased temperature usually increases growth, so why is it not a
Yes.
resource?
I know that's why I think it's just because.
I would say it's more of a condition. And it's not a resource because it can't be depleted.
Right. Yeah, that's the key thing is
Okay.
temperature, usually affects growth. But you don't like organisms typically don't change it too much. They don't consume temperature. So it's gonna be what can we call it? An environmental factor, perhaps?
So how about light?
Light.
How do you deplete light?
Yeah. All right. So we got a question here. How do you deplete light?
Well,
If you grow too much biomass, then you can cast shade on whatever it's under you.
Yeah. All right. So light stays on the list.
But in this sense,
Oh.
is there some like organism can absorb heat? I don't know.
I don't know. Not sure.
Change this condition, environment.
Right.
Well, I don't think to maybe affect other organisms, but definitely there's tons of things that absorb heat, ectotherms.
Okay. Nasser?
I don't know if this one might be a little out there, but I feel like for a lot of species that compete for like a good phenology, like time could be a resource. Like species, can kind of be depleted and you don't want to compete too much at the same time. So
Right. Yeah. So if we're gonna put space on the list, then we could put time on the list as well. Yeah. Because probably space itself is usually not limiting, right? You could always pack in probably more animals, more organisms. But it's like space is a proxy for all of the other resources that are kind of like in that same localized area. All right. So good.
Like
So I think we're all-
intertidal zones.
What's that? Okay.
Intertidal zones.
All right. There's always freaking ecology. There's always a counterexample. All right, so there are space limited systems.
That's like a classic one.
Yeah. All right. Okay, so we got some good ideas here. So how are we gonna model this resource competition? And remember, the goal here is to in some way simplify, but now we're gonna have to keep track of the resource, so it's actually gonna get a little bit more complicated before it gets simpler. So we're gonna couch our models today, at least, and often in class, in terms of this bit of laboratory equipment called a chemostat. And so chemostat is basically the equivalent from chemistry of a continuously stirred flow-through reactor. So it's a flask. You've got nutrients coming in. So there's a pump that's continuously pumping nutrients in. So the A is the rate of flow of nutrients coming in, and Rn is the concentration in the supply medium. And it's continuously stirred, so there's no spatial structure going on in there. And we don't want this thing overflowing and flooding the lab. So there's a drain, and medium goes out from the chemostat at the same rate, A. So it maintains constant volume in there. Okay, so this is the chemostat in the absence of organisms, right? So it's really not a lot going on there. So you can see we got an equation for the change in resource concentration inside the chemostat, and it's just the input minus the output. And you can just visualize at equilibrium, the nutrient level within the chemostat is going to match whatever you're pumping in, Rn. Okay, so this is kind of boring. This thing was invented in 1950, by Novik and I don't know how to pronounce them. I'm going to just say Shillard, who are biophysicists at University of Chicago. Don't know so much about Novik. The second author, Shillard, pretty interesting, like a physicist. He invented like chain reaction. So he invented like the nuclear chain reaction. And so he was there working on the Manhattan Project in the 40s, and then turned his and chain reaction is a form of exponential growth. So he was already like primed to move into ecology. So they invented this gadget in the 50s and even like figured out the theory of it. Okay, so oh yeah, other thing about chemostat. So yes, it's a real thing in the lab, but it's also kind of like itself not a bad model for some natural systems, right? You can imagine instead this is a lake and you got like streams coming in, bringing nutrients, streams going out. And it could also be like mixing across the thermocline in a stratified water column. It could also be, you know, for terrestrial systems, you have nutrients coming in from rain or application, and then, you know, they're leaching out the bottom. So this general setup isn't just restricted to the lab. Okay, so that's the chemostat. Now let's drop into species N1 and N2, who are going to use the resource and let them compete in this chemostat. Well, I should say, that's why I'm thinking about the chemostat. You know, we use a lot in the competition theory, but its original design wasn't so much for ecology, but more for physiology, because it's a way to maintain a culture in a constant, you know, well-defined environment. So it's growing at a constant rate. But okay, competition for one resource. So this is the setup. 3 equations keeping track of the resource and the abundance of our 2 species, N1 and N2. This first bit is just the abiotic supply and going down the drain. And then nutrients, well, I'm going to move down to the population equations. This stuff in parentheses here is the per capita growth rate of species 1. Okay, so then the total growth rate species 1 is its per capita growth rate times how many there are, N1. So stuff in parentheses, the per capita growth rate, we think about this a lot because it's kind of an individual based perspective, right? This is like what's happening to one particular individual. Okay, so growth depends on the resource R, according to some function that we're going to describe in a minute, mu1. Mu is kind of a traditional letter for growth. And so this is births minus deaths. And we're just going to assume here that the deaths are density independent and resource independent. It's just you die at rate m. And that death at rate m should include going down the drain in the chemostat. So it should include the A within the M, but here we're just going to give it a separate letter. And yeah, we got the same thing for both species. So that's what's happening with the N1s. Finally, looping back, when species grow, they consume nutrients. That's the other key thing. So this here is the uptake of the resource by species 1. And you see it's proportional to their resource dependent growth rate. But then we need a conversion factor, yeah? Because, well, it depends on what we measure N1 in, what units we measure N1 in. But if we measure the population in, say, cells per milliliter, and we measure nutrients in, you know, micromoles per milliliter, then we need to convert. And so that's what this Q is. And it's called the nutrient quota here. And so the nutrient quota is basically just how much nutrient there is per individual NI. Okay. And that's it. So this is the setup. Any question on what we got so far? Okay.
I guess,
well,
sorry,
yeah,
let me just
go on,
ask, I know like, in classic, or like when I think of like consumer resource interactions and like, I usually think of for the consumers having like a conversion efficiency. But I guess in this context, that only applies for biotic
All right.
resources.
All right. Sit tight. Wait for it.
Okay. all
Next
right,
slide. Okay, so usually, you know, when I'm usually teaching class, I just present it like one way or the other, usually this way. But there's basically two different ways you can look at it, and people do look at it two different ways. And so I just want to like mention both of them and show you how to convert between the two so you don't get confused later on, because I'm sure we'll run into both. So here we have a conversion factor, and it says nutrient quota, which is nutrient per cell per individual. But an alternate formulation that people use looks like this. So now instead of having this function mu1 up here, we have this new function v, which stands for, I guess, velocity of uptake. So now v is the per individual uptake rate of the resource. And then, so here we don't have the conversion factor anymore because we're dealing with uptake, but then we can't escape it. We need a conversion factor somewhere. So now we have the conversion factor down here in the growth equation. And now we'll call it Y, where Y stands for the yield, how much biomass you get per unit resource consumed. And you can see it's like the same thing, but just like, you know, inverse. So this is true. So the yield is just going to be 1 over the quota. So the same thing, just moving it around, put in different places. And on the right here is just showing that like the growth equals the yield times the uptake, or the uptake equals the quota times the growth. It's just different ways of looking at the same thing. And yeah, I guess one thing that you mentioned, Matt, that could also be mentioned here is that here we're assuming that like there's conservation of mass. So there's like no nutrients are lost in the process. If this were, you know, the energy of a biotic prey, then potentially some of that energy would be lost in the process. So then you'd have this extra sort of conversion efficiency in front there too. But here we're assuming it's some kind of nutrient like phosphate, and so that we'll assume is conserved.
But you can also incorporate that into the yield term. Into thinking about experimentally measure those levels.
Yeah, you could include in the yield term. You mean if like it's not perfect assimilation efficiency?
Yeah, if you measure the nutrient, like the nutrient they use and how much biomass is being built, the direct measurement would include the efficiency.
Yeah.
This energy.
Yeah. It's just the question is, if it's a nutrient and it's conserved, if you take it up, but then you don't use it to make new biomass, where does it go? Right here, it's like everything's like accounted for, so there's no loss. But anyway, so these are two different formulations people use, but they're interchangeable. You can always go back and forth. Yeah, Sarah?
Sorry, just to check up here, you were mentioning if you have the yield, so you take up the resource, for example, the phosphorus you were mentioning, but you don't make more biomass. Are you mentioning that the cell density would be increased, or does it also take storage in cells, for example, or this yield does not take internal storage into account?
Right. Yeah, good point. So this model assumes that it's just like there's no internal storage. It's just like you take it out and it turns into new biomass. And that's technically not true. So later on, at some point, I have to decide exactly when to slip this into the schedule. But later on, we'll look at a more complicated version where the nutrient per cell itself can vary. But as for now, just to keep it simple, we're just going to assume that like it's just instant, turn nutrient into new biomass at a fixed yield. Okay, so this is the setup. Now one thing left that we have to specify are these, well, either the uptake function or the growth function should be like proportional to each other. I'm going to draw it here in terms of the uptake function V, because then these functions V have a name in the ecology literature. It's what we call a functional response. So functional response basically tells you how fast you take up resources as a function of the resource concentration. Okay. And there are a lot of different ones people use. These are the two most popular. So on the first one on the left, we have the linear functional response, and it's just a line. Sometimes it's called type 1 functional response. These names, type 1, type 2, go back to C.S. Howling, who described them. Actually, there's some confusion in the literature too about exactly what's a type 1. Is it linear? Does it have like a top to it? I'm just going to say it's linear, okay? So linear relationship between uptake rate and availability of resource, and just characterized by one number, the slope. That's fine. Sometimes we'll do this in a model just because it's simpler, but it's not particularly realistic because at some point, you know how it is, right? You're trying to eat and like you have unlimited food available, but you're not going to eat infinitely fast. There's some limit to how fast you can consume. That's because of like your handling time, right? So you grab a cheeseburger, it takes you some time to unwrap it, eat it, chew it up, swallow it. And that time you're spent like dealing with the hamburger is time that you're not spending getting more hamburgers from the table. So that leads to the type 2 functional response. And so that goes by various names also. So Holland calls it type 2, but we also call it Michaelis-Menten-Monod. Michaelis-Menten comes from chemistry literature. Is sort of an enzyme, the rate of an enzyme catalyzed reaction. And basically it's the same logic, right? Because like, you know, an enzyme catalyzed reaction, your enzyme takes some time to grab the substrate and like deal with it. So it arises for the exact same reason, this saturating. But yeah, so it saturates. Monod comes from the microbiology literature. Okay, so how do we describe the type 2 functional response, Michaelis-Menten? And so this is how we can write it. So it's got two parameters. Vmax, that's your maximum uptake rate. So if the resource R were really huge, if we imagine let this R go to infinity, then V approaches Vmax. So Vmax, maximum uptake rate. And then we have to like have some shape parameter. We have to say like how fast do you reach the maximum, okay? But this is where it gets slightly tricky because you never reach the maximum, you just approach the maximum. So that's why they invented this thing called the half saturation constant. So you never reach Vmax, but you certainly reach Vmax over 2. And so we're going to say H is the resource concentration where your uptake rate is half maximum. And that's why it has the name half saturation constant. Okay. And so here is the algebraic expression for type 2 that we use. Sometimes people talk about uptake affinity. Uptake affinity is just the slope of this functional response at lower resource levels. So it's just the derivative of this evaluate at 0. So the affinity turns out to be Vmax over the half saturation constant. So affinity is some measure of how good you are at getting resources at low levels. Vmax is a level of how fast you are at getting resources at high levels. Okay. So that's the Michaelis-Menten. Any questions on that one?
Chris, is it like virtual equivalent to like writing it in terms of handling time? Is it like?
All
or
right,
there-
just for the record, Matt is not on the payroll here. I'm not sending these questions ahead of time. So alternative formulation. So Howling, wasn't interested particularly in nutrients and algae, for example, or bacteria. He's more interested in animals. And so when he derived the Howling type 2, he parameterized it this alternative way. And so he was imagining, was this the mantis? So he was imagining like insects going on looking for food. And so C is the attack rate, the rate that you're attacking hamburgers. And little h here is the handling time, how long it takes you to like capture, consume and digest, I guess, one prey item. So this is another formulation basically. And you can always scooch back and forth between them. So here I'm noting that the maximum uptake rate's 1 over the handling time. So you imagine if, in this case, if like food was saturating, you got all the hamburgers in the world are as infinity, then this would be basically C divided by CH. So basically the Vmax is 1 over the handling time, because it's like as soon as you're done eating one hamburger, you're grabbing the next one. So your uptake rate would be like 1 over the handling time. So these are all equivalent. Didn't Howling do something with undergrads where this is also called the disk equation. And the reason for that is because he took a bunch of undergrads and put blindfolds on them and had them hunting around for disks of sandpaper and parameterized it. So anyway, kind of random. All right, these are all equivalent. There are other functional responses that have the same form that maybe people use a different expression for, but doesn't matter a heck of a lot. Okay, so these are functional responses. And there's other ones, the Howling type 3, instead of increasing linearly at first, it's sigmoidal. So at low resource density, you forget how to look for food or something and it starts increasing slower. All right, that's all I got on functional responses. So any other thoughts on that? Okay, so we'll plug those into our resource competition model and let's see. So let's plot those out. Let's have 2 species. Here are their nutrient dependent growth functions. And then here I'm just gonna give them the same mortality rate. So they're gonna share the same M. Stick these 2 dudes in the chemostat and see who wins. So I'm gonna launch a poll for y'all and just tell me, who do you think's the better competitor? Okay, so I think we got most people here. So let's end the poll, see what people think. All right, so I can tell from the poll results, this may not be the first time y'all have seen this. So we got overwhelming support for species 2 winning. A couple people think species 1. A priori, if this is the first time you've ever seen it, I would look at it and be like, look, species 1 grows faster over a vast range of resource levels. Species 1 obviously fast, but that's the naive expectation, which will not turn out to be true. So let's check to see if this logic is right. So I'm gonna pop over to Mathematica now. There it is. And All right, so here it is. So hopefully this works out. Load package. You know, I started getting this error message. It's more of a warning. I think Wolfram started using my word set model. So it's like causes this little warning message. So I have to change the name of set model, I think. It'll work fine. It'll just be red here. So I set up same thing we had before. So we've got the two species are going to be green and dark green. Resource is going to be blue. It's just the same competition and chemostat equation we had before, with the type 2 functional response with the maximum growth rate and a half saturation constant. So we can assign parameter values and plot the growth versus resource and the mortality, and same plot we had before. And now to see what happens in competition, we'll just stick them in the virtual chemostat, simulate it for 200 time steps, and see what happens. Okay, maybe I'll make this smaller so we can keep track of what's going on. Okay, so this is the dynamics of competition then. So top graph shows the abundance of the two species, bottom graph shows the resource availability. So we're starting at 10, which is what we're dumping into the chemostat. And they both like that. Both growing exponentially at the beginning. And light green, that's species N1, who we would naively think should be best, gets off to a head start and is doing great. So it looks like species 1 is going to win. But unfortunately, it peaks early and then plummets. Meanwhile, species 2 kind of slow going, but keeps growing and kind of approaches its carrying capacity, let's call it. So it looks kind of like a logistic growth for species 2. And we can understand what happened from looking at the resource graph. So we're going to consume resources down as populations grow, and then we're going to hit this level, this dashed line here. What's this dashed line? This dashed line is this point here. It's the resource level where species 1, its birth rate just equals its death rate. Okay, so there's going to be a key value here. So at this point, species 1, light green, is just breaking even, births equal deaths. But species 2, still feeling kind of okay, right? Its birth rate still exceeds its death rate at that point. So species 1 is going to peak here, but species 2 keeps going. As species 2 keeps going, it's going to draw resources down to this point here. At this point, species 2 is just breaking even, zero net growth. But unfortunately for species 1, its birth rate now is less than its death rate. So species 1 is on its way to competitive exclusion. And so we can see from this that the key factor, the number that determines the outcome of competition, is this break-even nutrient concentration, where, let's switch over here. Okay, so this is the what we call the R-star rule. So each species can be characterized by a resource level where its birth rate just equals its death rate. We'll call it the break-even resource concentration or zero net growth resource concentration. And then the rule of competition for one resource is whichever species has the lowest R-star will outcompete the others, all others, okay? And these expressions on the right are just looking at the algebraically, the expression for R-star, our measure of competitive ability, as a function of these, more basic physiological parameters. So the mortality rate, half saturation constant, maximum growth rate, mortality rate. And we could also break up the maximum growth rate into the yield and the maximum uptake rate if we want to think of it like that. Okay, so that's the outcome of competition for one resource. Coexistence, mostly impossible, right? The only way we could get two species to coexist on one resource is if they had equal R-stars. And you can imagine that we could do that. So if we can, suppose we could manually adjust the mortality rate of the two species. If we adjusted it so that the mortality rate of the two species would be equal, then they would be coexisting, but they'd be coexisting in kind of a neutral sense. So let's just try that. Ignore this a little bit. So here I'm just solving for where the two growth curves are equal and looking at that point, 2/3. So according to this, if we make the shared death rate 2/3, then they're going to have equal R stars and therefore coexist in a neutral sense. So let's try it. Okay, so we see here, got equal R stars, and well, it looks like light green is doing really well compared to dark green, because light green, you remember, is grows fast at high resource abundances, so it got a good head start. But the key thing for dark green species 2 is it's still there, right? Its abundance at the end is 1% of species 1's abundance, but it's not 0. Okay, so this is actually neutral coexistence. And if we were to increase like the initial abundance of species 1, then we can see that species 1 now looks better in the long term. So that's kind of the signature of neutral coexistence, is that we change the initial conditions and we change the end result in a sort of continuous fashion. So this is the only way we can get coexistence, and it's a little bit precarious because they have to be perfectly balanced. So if we change this just a little bit, make the dilution rate a little bit more than 2/3, let's say 0.7, try that. Yeah, then we see that now species 2 is on its way extinct here. So anything else here? Yeah, so that's the R star rule of competition for one resource. Any last thoughts on that?
Yeah, this might be kind of a contrived example, but is there a situation in which the difference in growth functions, there's a difference in growth functions, but you still have coexistence between two species because their death rates are different? Like somehow it's like offset by their death rates. And so they actually have like, for a given resource level, they actually have like equivalent fitness because one is just dying faster than the other.
Yeah. Here I was kind of illustrating it with two species that have the same death rate. But you see that if we have species dependent death rates, as long as we can cook up a case so that their R stars are the same, then it'll be this kind of neutral coexistence. And we could do that by tweaking the death rates as well.
Okay, got it. Thanks.
Okay, so this is cool. We can measure all this stuff in lab. It's predictive, mechanistic model. The downside is, compared to Lotka-Volterra competition, now we've basically got 3 of the Lotka-Volterra outcomes. We got species 1 wins, we got species 2 wins, we've got neutral coexistence, but we don't have proper coexistence in terms of mutual invasibility, and we don't have founder control showing up here either. So this is a consequence of the fact that there's only one limiting factor, one limiting resource, and the R star rule. So to get around that, we're just going to have to add a second resource. Is probably the easiest way. And so this is the second main topic for today, competition for 2 resources. This is showing the equations kind of in generic form. Notation gets a little bit heavy because we have to have these conversion factors for 2 species and 2 resources. So that's the QIJ. So that QIJ is the quota of species I on resource J. And then we're going to have to sort out this growth function mu I, because mu I now depends not just on one resource, but on both of them. And so how do we put those 2 together? So this idea of 2 resource competition goes back at least to MacArthur. who looked at in his book, maybe even MacArthur and Levins before that. This general theory, like usually we kind of like ascribe it to Dave Tillman because he was a major proponent of it, tested experimentally and like really like built his early career on it. But actually, this is a sleeper of a paper there, but y'all got to check it out. Leon and Thompson were first. So Leon and Thompson, Journal of Theoretical Biology, 1975, basically worked out all the theory of competition for two resources in pretty clear detail. So don't forget those guys. All right, so what is this competition for two resource theory? Now we need to think about, since we've got two resources, we have to classify nutritionally what kind of resource pairs we're dealing with. And there's a lot of different varieties. The big two that we're going to focus on are what we call essential resources or substitutable resources. Essential resources are things that you need them both to grow and live. So for humans, it could be proteins and fats. Okay. I would normally say proteins and carbohydrates, but I think you could probably lose the carbohydrates and just live off protein, but you got to have fats too. But it's easier to think about these for plants or phytoplankton. So for plants, essential resources are things like nitrogen, phosphorus. Yeah, you need them both to live. And so the way we model essential resources, well, of course, there's plenty of different ways, but a super common way is using this Liebig's law of the minimum, which says that your growth, on two resources, depends on whichever one is in short supply. So these functions are the single resource dependent growth functions, and then you just calculate them and compare whichever one is smaller is what limits your growth. Of course, there's like interaction between the two and it could be more complicated, but this is the simplest. And then the opposite example is what we call substitutable resources. This one's easier for humans. That's like chicken and turkey, right? They both satisfy the same nutritional needs, and your growth rate, when you eat the two of them, just sum up your growth rate on each one by itself. So these are the simplest cases. Obviously, it could be more nutritionally complicated than that.
So the inner storage mechanism would also change this growth rate function.
Yeah. If we get to like stored nutrients, then we'll have to think where we put this function, basically. Any other question about types of resources? So you can't really talk about like a particular resource, whether it's essential or substitutable, because it sort of depends on what resource you're comparing it to. So nitrogen, for example, everyone needs nitrogen, but plants can take it up in different forms. Plants, at least phytoplankton can. Nitrate, nitrite, urea, ammonia, right? So those are like 4 substitutable forms of nitrogen, but you can't substitute any of them for phosphorus. So okay, so we're gonna really delve into this case of essential resources because following Tillman basically, and because a lot of us work on autotrophs. So just think of the 2 resources, nitrogen and phosphorus. All right, so the cool thing about resource competition theory for 2 resources, like if we had to do it mathematically following Leon and Thompson, we'd have like 4 equations. It'd be a pain to solve for equilibria, and Jacobian matrix would be 4 by 4. No one wants to find those eigenvalues. But luckily there's a graphical approach that tells you the outcome of competition and also promotes intuition. So the graphical approach for 2 resource competition, we're going to work in the plane of the resource availability. So R1, R2 plane. And the reason we can work in the R1, R2 plane here, and we don't have to worry about the ends in particular, is because of this growth function. So in brackets here, we've got the net per capita growth rate of species I. And you see that the per capita growth rate of species I only directly depends on the resource availability. So the per capita growth rate only depends on R1 and R2. That means we can work in the R1, R2 plane. So if there is some direct interaction between species, then it's going to be like, we can't do it like this. So this is one of the nice things about resource competition. Okay, so the first thing we need, we're going to have 3 ingredients for this graphical approach in the R1, R2 plane. Ingredient 1 are 0 net growth isoclines. So for any given point in here, for a combination of R1 and R2, the net growth rate, birth minus death, is either positive, negative, or 0. And so up here in the upper right, got lots of both resources. Birth rate exceeds death rate, so the net growth is positive here. Down underneath this curve, one or both resources is limiting, don't have enough of it, so the birth rate's less than the death rate, so net growth rate's negative. And so then this border between them is the set of resource availabilities where births just equal deaths. In the case of 2 essential resources, coupled by Liebig's law of the minimum, it's this L-shaped 0 net growth isocline, we call it, or ZINGI for short. Okay. So ingredient 1, 0 net growth isocline. Any question on that thing?
Chris, it's would be the exact same like graphical representation even if you had a consumer. It's like nothing is different between like the consumer resource, like Zingi's and representation of this, or is there?
Well, this is kind of looking at one species in isolation. We'll put 2 species together in a second. Okay. So this is kind of like a slice through the full phase space, but because the per capita growth rate doesn't depend on N, then we can get away with what we're doing basically. All right, so Zingi's. Next thing we need, so Zingi's are kind of measuring your response to the nutrients. Now we need the flip side, we need to know how you're consuming the resources. And so that's the next ingredient called a consumption vector. In this model, and often we're just going to assume that you consume resources in a fixed ratio. So you consume resources in the same ratio that you require them. So the ratio of your nutrient quotas for the 2 nutrients. So this arrow says that when you grow, this is how you're pulling down the 2 resource levels. And here we're just going to assume it's fixed. And in this particular model, we're going to assume it doesn't depend on the availability of the resources. Okay. And finally, we need to know something about the environment. So, and here the environment is going to be characterized by the resource availability that you're dumping into your chemostat. So RN1, RN2, that's the nutrient concentrations you're pouring into the chemostat. Okay, so those are the 3 ingredients that we need here. So for a single species, the way I like to draw it is, you can imagine starting out with not a lot of biomass. Nutrient supply matches what you're nutrient availability matches what you're dumping in. As the population grows, they consume resources according to their consumption vector, what the ratio that they need them. And that's all well and good until it runs into the 0 net growth isocline, at which point births just equal death. And then you reach some sort of equilibrium. Okay, so this is, you know, a cartoon for the dynamics of a single species. But now we want competition. But wait, any question on this single species thing here? One thing I could say, add here, that like where you end up on the zero net growth isocline tells you something about what resource is limiting. So in this particular case, we ran into this horizontal part of the zero net growth isocline. That tells us that resource 2 is limiting at equilibrium. Because you can imagine if we add some resource 1, move up, then growth becomes positive. But if we were here and we added some resource, wait, did I say 1? If we add some 2, growth becomes positive. If we add some resource 1, you're still on the zero net growth isocline. Didn't help because resource 1 is not limiting because that minimum. So down here, we'd say resource 2 is limiting. If we end up over here with a different supply point, then we say resource 1 is limiting. And if you happen to end up at the corner, then you're equally limited or co-limited by the 2 resources. Okay, so now let's put in 2 species. And let's assume that there's a trade-off between compatibility for resource 1, resource 2. One thing I should say here, I forgot to mention, this zero net growth isocline, just think of it as kind of like, it's like the multi-resource version of R star. Okay, it's where births equals deaths, but now it's a function of 2 resources. And for essential resources, this corner here is kind of like the R star on resource 1 alone, R star on resource 2 alone. Okay, so then if we put in 2 species like this, these 2 species, there's a trade-off in their compatibility for the 2 resources. So if only resource 1 was limiting, then we see that species 1's R star for resource 1 is lower than species 2's. So if it's all about resource 1, species 1 would win. But conversely, if it was resource 2 is limiting, then we see that this point here, species 2's R star for resource 2 is less than species 1's. Therefore, if his resource 2 is limiting, 2 would win. And so we see that there's a trade-off in their compatibility based on the fact that these 2 zingies intersect. And since they intersect, that means there's one special point in this whole R1, R2 plane consistent with both species still being alive at equilibrium, which is this. So we call this a coexistence point. So at this one coexistence point here, both species have zero net growth and therefore are persisting. Okay, so at least some potential for coexistence here, but whether we actually end up there, let's see, because this is just one point. Maybe it's going to be another case of that neutral stability, right? Okay, so we can graphically sort this out using invasion analysis. So I'm going to take those consumption vectors at the coexistence point, plot them at the coexistence point, and then kind of extend them back on this diagram here. And let's imagine a supply point over here to the left of this little wedge, okay? So if we put the supply point here, let's do the invasion analysis to sort out the outcome of competition. Put in species 1 by itself. It's gonna consume resources according to its consumption vector till it runs into its zero net growth isocline. Here, this is species 1 monoculture equilibrium. And then we sprinkle in some species 2 and see if species 2 can invade. Well, this point here, this equilibrium point with species 1 alone, it's below species 2's zero net growth isocline, which means that 2 can't grow there, right? So therefore, species 2 can't invade species 1. Now, if we do the opposite, put in species 2 by itself, it's going to quickly become limited by resource 1. But once it ends up there, ask if species 1 can invade. It can, because this point here is above species 1's isoocline. Therefore, species 1 can invade 2. So if we put those two things together, then we see that 1 can invade 2, 2 cannot invade 1. That's the outcome, species 1 wins. Okay, so for this particular environment, for this particular supply point, species 1 outcompetes species 2. Any question on this invasion analysis concept? All right, well, let's do the same thing now, but let's just change the environment. Let's move the supply point into this wedge here and see what happens. We can do that in the chemostat just by mixing up some different medium. So now we have a more equitable supply of the two resources. Put species 1 in by itself, consumes resources down to here. Now this point ends up above species 2's thingy, so 2 can invade 1. Flip it around, put in 2 by itself. It's going to consume resources until it's limited by resource 1. And then resource 1 can invade it. And so basically, if we put those two things together, we see for this particular supply point, the two species can stably coexist. And when I say stably coexist, I mean, by way of mutual invasion. Okay, so this isn't like some kind of neutral stability. This is like proper coexistence, like in lac of Volterra. And, you could convince yourself based on sort of how these lines here are parallel to these borders, and it was all driven by the consumption vectors. This is going to be true. This outcome of species 1 and 2 coexistence will be true for any supply point in this wedge. And if we go down here, well, then 2 is just going to win, right? So we can combine these outcomes on this plot here. And this basically gives you a map of the outcome of competition as a function of the supply point. So over here, if we're dumping in lots of resource 2 compared to resource 1, then 1 becomes limiting, and species 1's the better competitor for resource 1, so 1 wins. Opposite down here, but in between, for these intermediate supply points, somehow between the two of them, we get sucked into this coexistence point, and therefore the two coexist. So yeah, this is the outcome dependent supply point.
Chris, I got a question.
Okay.
So with this assumes, so with an invasion analysis, a species is like reaches its carrying capacity, and then like we're assuming that kind of dynamics where a species reaches its carrying capacity, we just add a little bit of the other, and then it's able to. Okay, this is just okay. This is a little trippy for me.
Yeah,
I don't know why. Just because I feel
like
compared to the lock of, I don't know. Okay, yeah, I learned all this stuff a long time ago.
Yeah, I mean, the invasion analysis is pretty handy, right? Because it like makes it easier. We can like sort out the outcome of competition just using these like arrows on this graph here. With invasion analysis, we didn't have to like calculate any like 4-dimensional Jacobian matrix and so on. But like, we talked about a few times ago when we introduced invasion analysis, when it works, it's great. It doesn't always work. There's like these weird borderline weird cases where it doesn't work. Be on the lookout for those cases. But I can say in this case, it does work. There's no weird stuff going on here.
Yeah, I guess it just makes me think so I guess the key, the main difference here is that it's more of that, so who wins in competition is determined by environmental conditions, whereas in I feel like in the lac of Volterra, it's just more by your interaction strengths, which I guess now I'm like,
yeah, I mean, so here, I mean, this is kind of like, this is like, if we go back to that checklist of problems with lack of Volterra competition, this solves a lot of those problems, right? Because
Yeah,
like
yeah.
now we can see that like we can measure, the components of the zero net growth isocline in the lab. We can measure the consumption vectors on species by themselves in the lab. And now we can sort of map out how the outcome of competition depends on the environment, where the environment here is the, supply ratio of the two resources, where in lack of Volterra is kind of like environment, what environment, right? You can't really see.
Exactly. Right.
So
That's
this is really the, this is like getting at the heart of, why the resource competition theory is kind of useful. All right, so now we got like lack of Volterra outcome number 4, coexistence, but there's that last one, the founder control. Whoever gets in there first locks up the system and outcompetes the other. Can we get that in the 2 resource competition model? And the answer is yes. The way we're going to do it is we're going to take these consumption vectors and just flip them around. So here's the opposite case where now instead of having stable coexistence in the middle, so if we add the supply point here, put in species 1 by itself, it's going to consume resources down till it's limited by resource 1, and that's below species 2's isocline, so 2 can't invade 1. But if we put in 2 first, 2 would consume a lot of resource 2 until it became limited by resource 2, and then species 1 can't handle that, can't invade. So this is an example of the founder control. So the way we get founder control here is by just changing the order of these consumption vectors so that you consume more of the resource that you're the better competitor for. You're kind of a jerk, right? That's like the rule of founder control. You got to be a jerk. You have to like consume a resource and you're like, yeah, I don't really need that resource. The other guy does, but I'm going to consume the heck out of it and keep him out, right? And they're both doing the same thing. So whoever gets in there first locks the other one out. Okay, so this is how we get founder control. Yeah, so this is right. This is the mapping the outcome. So we still have these regions of dominance, but instead of being like a wedge of coexistence, a wedge of founder control. So let's summarize what this tells us about what it takes for species to coexist. There are going to be 3 things that we need. First of all, we need the zero net growth isoclines to cross. So there needs to be some trade-off between competitive ability for the 2 resources. And we can think of that, and then we see that graphically through the zero net growth isoclines. So that's a trade-off in the resource requirements. Second, we also need a trade-off between the impact measured by consumption vectors. So you have to consume more of the resource that limits yourself to get coexistence. And finally, it also depends on the environment, which is characterized by the supply point here. You need to have the intermediate supply point to get coexistence. So these are kind of like, intuitively how we can understand the requirements for coexistence. All right, well, let's pop backward to Mathematica really quick and see if this actually works out like this. Okay. So here's the 2 resource competition model. This is just some little point we're going to need each of the intersection, the coexistence point. We can assign some parameters, and here you see in the quotas we're putting in the trade-off now between the 2 resource consumption vectors. So species, how do I have this written? Species, yeah, so this is species 1. So species 1 consumes more resource 2. Species two consumes more resource one. But there's also a trade-off in their growth functions, these mu's. I'm using type 1 functional response to keep it simple. So, okay, so we can calculate the R stars, and the R stars show that species 1 is the better competitor for resource 1, and species 2 is the better competitor for resource 2. And then just plot, here's our little resource phase plane, and we can see that this is the case of coexistence if we slip a supply point in here. Yeah. So now we can numerically solve. So let's pick a supply point in this coexistence region, but not quite symmetric, so we can see what's going on. Pick supply point 3.1, 3.0. Initialize the system with low abundance of competitors and let them go at it. Okay, so this is the time dynamics of the two species, and they're both doing fine at the end. Species 2 is a little bit more abundant, but that's because of the slightly asymmetric supply point. And this is showing how the resources are consumed. So we start at the supply point, they consume resources down, and they get drawn into this coexistence point, and then they're all happy, coexistence. This last bit here is just looking at the last point, what's the abundance of all the two species, two resources. And finally, just to verify that all that Jacobian matrix stuff is true, this is the eigenvalues corresponding to this final point that they end up with, and they're all negatives, so it's stable. Okay, so that's stable coexistence. Now we can change the supply point within the coexistence region. We can crank it up to the right a little bit. And so now we move the supply point to the right. Now you see that they still coexist, but now we're ending up with more species 2. Yeah. But they're still happy, and the system still ends up at this one intersection point. If we take the supply point and move it down, let's say down to down here, then we should be getting into the realm of competitive exclusion. Yeah, so now we see that species 2 wins because the supply point's outside the coexistence region and so on. So I'll send you this if you want to play with it, but yeah, works like we figured out based on drawing lines. What's this? Can we change now? Right, so now we want to change it. Oh, this is just a little riddle here. Can we change species parameters to show the founder control case? Okay, so let's try it. Let's go up here. So it should be that if we change the quotas, that we could get into the founder control case, maybe. I'm going to change, just flip these around and see what happens. Okay, so that actually worked out. So according to this, now it's in the zone of founder control, and we see that because this is just a little trick that I used to teach my students when taking exams. If you go around this coexistence point in a circle, and if you get two of the same color in a row, then it's founder control. They alternate, stable coexistence. That's the trick. So now if we move the supply point back into this center and move it right in the center, and we're going to give species 1 a little bit of an advantage at the start, just a little bit. Okay, so now we see that indeed species 1 wins, and we start out approaching this coexistence point, but then we take a sharp right and end up at species 1 winning. But if this is truly founder control, then if we just change the initial abundance of our two species, we're not changing any of the parameters, just initial conditions. Now we should get species 2 winning. All right, awesome. Okay, so this is the founder control case. Rest of these I don't really want to get into because not too interesting. Any question on those simulations? All right, so it matches what we intuited from the graphical approach. Yeah, one other thing I want to point out. So this model makes predictions about how species composition or community composition will change along environmental gradients. So Tillman sort of popularized this under the name of resource ratio theory, with the idea that the relative abundance of resources varies in nature. So if we change the supply point along this gradient here from large R2 to R1 ratio down to small R2 to R1, then we should get a change in the outcome from species 1 wins, coexist to species 2. And in the coexistence region, as we're taking this slice along here, you can see that the abundance of species 1 declines, whereas species 2 increases. So we get some predictions about the relative abundance of species along this environmental gradient in the coexistence region. So they coexist, they'll be happy forever, but one will be a little bit more happy than the other, depending on the supply point. All right, so that's it for essential resources. Substitutable resources works out the same way, more or less. In the case of substitutable resources, where we add up, your intake from the two resources, the zero net growth isoclines turn out to be lines with negative slope. But again, if these cross, there's a coexistence point, and if you set up the consumption vectors properly, then it'll map out this coexistence region. So it works out pretty much the same with substitutable resources. So any question on this basic two resource competition theory? Yeah. Yeah, Matt?
Yeah, I guess this might be a subject for later, but I guess it gets more complicated if the resources are biotic.
Yeah. Yeah, right. So this all this works out, the graphical approach works out perfectly in the case of chemostat kind of resource. And so you're talking about biotic resources. So here, this really works best for resources that are not alive and are being dumped into the chemostat. For living resources like prey or plants or something, then instead of having that chemostat supply equation for the resource itself would be alive and maybe has its own logistic growth. Yeah.
So that was okay. That leads to my other question, but
okay.
Yes. I guess if let's say you put just one resource, one consumer in the chemostat with the resource, would it exhibit logistic growth?
It'll exhibit something similar to logistic growth, not identical, but so would it be exactly logistic growth? No. Would it start out growing exponentially and then run out of resource and kind of run into a carrying capacity? Yeah.
Okay. So then my other question is do you have any good, I don't know, because I'm really interested in this subject of for competition in the case of biotic resources, if you want to do more of a mechanistic resource competition, what is a good reference or a starting point?
Yeah. We'll probably bonk into that at some point.
Okay.
So I guess what you're saying here, but just so everyone's on the same page here, what Matt's saying is that suppose this resource were itself alive, then this wouldn't necessarily be the right equation for its dynamics by itself. We could replace this with, say, logistic growth, which would just have an extra little R out here. A lot of the conclusions will carry over regardless, because this actually didn't get into our argument about the R star rule. So the R star rule was based strictly on the growth equations of the two consumers. So what the resource is doing on its own time doesn't factor into the outcome of competition. So the R star rule basically is going to be identical. If the
So it's just like replacing the chemostat like dynamics just with logistic.
Yeah, we just plug that
Then
in there
it's all-
and, the expressions will be a little bit different, but the outcome of
Yeah.
competition will still be like determined by R star. The, for two resources, probably similar also. Maybe some of the details will differ with, mapping the supply point into the outcome. But yeah, basically not too much.
Okay.
Not too much.
Okay.
It's pretty good.
Okay.
We'll run into that at some point. So, all right. So one last thought before we go, which is going to set the stage for a lot of the rest of the semester here. So we've got around competitive exclusion by introducing a second resource, and I was like, yeah, got two species coexisting if meet the right conditions. What if there's more species than just two species out there? Could we get three species that coexist in this case? And the answer is no. So if you have two resources and we plot like the zero net growth isoclines for species 1 and 2, and like let's put in intermediate species 1.5, who's kind of like in between, maybe we could get the three of them to coexist. But no. To coexist, we need a point in this R1, R2 plane where all three species have zero net growth. And there's no such point, right? In general, unless you fine tune it somehow and get lucky, you're not going to have three curves on this plane intersecting in a single point, which is what would be required for coexistence of more than two species. So this is basically the logic behind what's been called the competitive exclusion principle. And I'll just call it competitive exclusion principle version 1.0 because we're going to be like applying some patches later on. So this is no more species can coexist than there are limiting resources. So if you got one resource, one species max. If you have two resources, two species max, and so on. And so this gave rise to what G. Evelyn Hutchinson called the paradox of the plankton. He basically pointed out that phytoplankton, they compete just for a handful of limiting resources. Well-mixed systems in lakes and the ocean, but nevertheless, we get a lot more species coexisting than there are limiting resources. That was the paradox. And so that basically set off a whole lot of work in ecology trying to solve Hutchinson's paradox of the plankton, come up with these coexistence mechanisms. So that'll be like kind of a, classic introduction for a paper on modeling competition. That'll be kind of like the thread we'll follow throughout the semester here. How to fix this competitive exclusion principle and get more species to coexist. All right, so that's all my slides. Any last questions? Thoughts?
Who does that? Like the founder control? What is an example of things using resources that they have for quotas?
Yeah, we'll have to, we should look and see if we can find like a concrete example of the founder control. I'm sure they exist.
Right.
But yeah, species have to like consume resources they don't really like need. So they need to waste their energy consuming resources they don't need. So I think it's less likely outcome. So I don't think it's like totally even. I think nature will be predisposed more towards coexistence than founder control, but we'll see.
Chris, I have one other kind of comment question. I guess I think it's more of a question. So would you say that like Locke-Volterra, not Locke-Volterra competition, but if you treat like if you use Locke-Volterra equations for the resources, and then you have two consumers that are indirectly competing through those shared resources, then that is totally consistent with resource competition theory.
You're saying the resources themselves are competing according to Locke-Volterra?
No. It's kind of like no. They don't compete. They they have their own like logistic growth.
Yeah, so basically you're just saying like swap out the chemostat equation with logistic growth through the two prey?
Exactly.
Yeah.
And yeah, it's
Yeah, I
yeah.
mean, I think most of what we see will carry over.
Okay, gotcha.
Yeah. But we'll probably run into that at some point too. All right. Well, thanks y'all. Next time I've got some papers where people are like looking at the empirical evidence for this resource competition theory. So I think that'll be a nice thing to discuss for the first half. And then the second half, I haven't quite decided which direction to go. Maybe more physiological reality, maybe looking at those biotic resources and substitutable resources more. Maybe looking at positive interactions. We'll do all those things at some point, but which one we're doing next week, I haven't quite decided. So stay tuned for that. All right. See y'all next week.
Thanks, Chris.